time
Oh,
I
this
showing
okay.
you was not
I
there.
didn't get this one. I didn't get this
So
paper
you
yet.
know, for the two marker question, look, 1, 2, 3, 4, 5, 6. So six words are there.
Yes, sir.
And 6 4 ja 24. And okay, 4, 5, 6, 7. So almost it's a 40 45 words are over there. This is good enough, right? 45 50 is good enough. So for two marker it was good enough, right? And the things are, the content is there, right? So
Yes,
content
sir.
means sometime, you know what happens, sometimes student try to beat about the bush.
What about my sixth answer, sir? Was it not too sure?
Look, like in this in this answer, suppose that if you will for the two marker question, if you'll write it down at your school, you won't get 2 marks. They will give you hardly 1 mark. I was a bit liberal.
Yes, sir.
So content should be over there. So if they are, so how much you written over there, how what was the content, right? How much content did you give? What
Sir,
was the word in three
I'd
marker question?
say I wrote around 80 words for the 3 mark question on
80
average.
words. I think the number of the words were sufficient. Then what teacher said that content is missing over there?
She said
Words.
it wasn't specific enough.
Okay, it was not specific enough. Can you share your
Yes,
question
sir.
paper with me?
I didn't get that, sir. She just announced the marks in class, although I can show you the test that I did recently.
Okay, write it down some questions and give me some question, and I'll tell you. I will
One
try to know
second, sir.
what you have written over there, right?
Uh,
And I will try to know
so should
that.
I send it on WhatsApp?
Yeah, you can send me the question paper, your question.
Can you just let me know your number?
Yeah, sure. Write it down.
Yes, sir.
Uh, Add on +91, right? Eight three seven
Yes,
six.
sir. 8,
8376.
yes,
0
yes.
42
Okay.
6 695.
695.
Right. Share the question with me, right? And I
Okay,
would
sir.
like to
I'll
know.
I'll share the class test we did 3 days ago, where I got 5 out of 8.
Sure. Sure.
Okay, sir. Just one
Yeah.
second.
Yes, Martin, what we did in last class. Martin, hello. Martin, I am unable to listen you. Are you there? Martin, hello, Martin. Are you there? I'm unable to listen you. Open your
Yes,
mic.
sir.
Okay.
Yeah,
What we did in last class, Martin?
yes, sir.
Did I dictate
Water potential. We're learning about water potential,
water potential? Okay. So I
plant
think
transport.
water potential. Did I dictated it?
Yes, sir.
Okay, fine, fine. So we have to start from here onwards, right? So, Shidika, are you sharing right now?
Yes, sir. I'm just taking my paper out. I added your name.
Martin, look, we are discussing that Shidika, she written the question paper, and her teacher complained that content is missing over there, right? And that's why every time I poke you, like you, it doesn't mean, it doesn't matter how smart you are. It doesn't matter what content is there in your mind. But you know what actually matter when you are in examination hall, that what is like your representation power, right? How well you are representing yourself in terms of your answer writing, right? So the content is missing, right? And your answers content is missing, you write very less, and it should be more legible and clear. So what was the question? Yes, Siddhika, I got it. Uh,
Yes, sir.
so,
This was my test
so
from a teacher.
he has given you a 5 out of 8, right?
Yes, sir.
And your question was, the question was, explain the life cycle of Teridophyte. Was it there? No, Bryophyte. You did. Then the life cycle. Okay. Uh, explain the life cycle of Bryophyte. 3 marker.
Sir, I did all three questions. The pteridophyte is in the second page.
You did all 3 questions in the same page. That is
No, sir, it's on the second page.
second page. Okay. So this. Okay. Yeah, you have shared one more page. Fine. So Pteridophyte. Look, first thing what is missing, diagram is missing. You must represent the life cycle. Look, whenever I always ask you people to write down, draw the life cycle. I think the plant kingdom should be here. Plant kingdom.
Sir, for
Uh,
this specific test, she had told it wasn't necessary
life
because it
cycle
is
of
just such a small test.
life cycle of Bryophyte. You have to, at least you have to draw the, like, life cycle of in life cycle of Bryophyte. You can do one thing. Look, where is that Bryophyte? Look, this one. Mhm. At least you should
Okay,
draw
sir.
this diagram. Bryophyte, right? Gametophyte,
Yes, sir.
sporophyte, germination, right? Zygote, egg, sperm, gametophyte. So at least you can draw this one, right? This is schematic diagram.
Okay,
First, first thing which was missing. I always say, whenever if they have asked for a life cycle, you have to draw at least a schematic diagram. If you can draw the, like, small bryophyte, then if you can draw the small diagram of sporophyte, that will be the best thing which you can do. At least what you could have done, you could have drawn your gametophyte, a sporophyte, and a small circle of for the zygote. That was sufficient enough, right? And
yes, sir.
bryophyte are also known as the plant kingdom, because we can live in the soil and sexual reproduction. The main body of the bryophyte consists of the main body of the bryophyte is consists of the gametophyte, which produces its own uh, germ cells, anthelia, and the production division. Instead of these two living the structure on photosynthetic. Uh, Gametophyte and derive the nutrition from the some of the cells are the reproduction division, meiosis, germinate into the spore of the different type, and these spore into a new body known as the sporophyte. Look, and the question is, like, your answer is, like, if they are saying the content is missing, I won't say that too much content is missing over there. Right.
Yes, sir.
Almost you have
Um,
put down this 70% to 75% information you have put down in this question, right? As per the question,
yes, sir.
right? The only thing which is missing, that is you should draw this kind of the flow diagram, right? And some diagram
Yes,
you could
sir.
have drawn, right? And you have to divide what is missing, right? You have to divide the question in different parts. First, bryophyte, you have
Okay, sir.
introduced the bryophyte right way, right? And along with that, you could have, rather than introducing like this way, you could have drawn that bryophyte. The life cycle of the bryophyte is, there are two different kind of the physical individual: gametophytic and sporophyte, right? And zygote as well, right? Then draw the
Yes,
diagram,
sir.
and this is schematic diagram. And after that, you could have done first paragraph, there should be, after introduction, there should be one small paragraph that should have been dedicated to gametophyte. Then change the paragraph, next paragraph.
Okay, sir.
Or you could, and for the sake of school examination, I don't know what they prefer basically. It depends on the preference of teacher at
Yes,
your
sir.
country. But the first introduction, then you could have drawn a small heading, gametophyte. Then a small heading, sporophyte, right? Zygote, and then the sporophyte,
Okay, sir.
and different three. And nothing you need to do. You need to draw only a small diagram. Gametophyte, draw the diagram like this way.
Okay, sir.
That's it.
Sir,
Got it.
about the third question, On
Yeah,
the third question, my teacher had said we need at least 4 points for 2 marks.
yeah, yeah.
Wasn't my answer sufficient?
Your
She
answer was
told me
sufficient,
just
but
one.
she or he need not to give 5 marks. If you would ask
No,
me,
no, the third question was 2 marks. And my teacher had said that for like for 1 mark we need 2 points. And I
for
wrote
one mark,
for
for 1 mark,
yes
we
sir.
need 2 point. Yes, yes.
Yes sir.
For 1 mark,
And
you
I
need to
wrote
write at
four,
least 2 point.
but she gave me one.
So what was the
And I'm
weightage of
just
this question? What was the weightage of this 3 marker, right? Out of 3 mark, how much he
no
has given? 2 marks. Yes, yes, yes.
no
You
for
know,
the third question. Differentiate between microsporophyll and metasporophyll.
yes, yes. Then I said in the initial in the starting that for if they are asking for difference, right? There should be if it
Yes?
is 3 marker question, there should be at least 6 difference, 6 point.
Okay, sir.
Yeah, yeah. That's what I said. So yeah, even in this difference, I will give you the 1 mark.
Okay, sir.
Right?
Okay.
But rest of your questions, rest of two answers, uh, content is sufficient enough, right? What you could have done first is schematic diagram like this way, what I told you already. With the help
Yes.
of this schematic diagram, I always got very good marks till my master's class. Always got very good.
Uh,
Uh, you know,
okay, sir.
these are the way to take the marks. That's why I every time I emphasize, and every chapter you will get the schematic diagram. I always represent it, right? I always teaches it, because they are very beneficial to take marks. They are not important uh, for your competition and all that, but they are very important for your school examination.
Okay,
What do you
sir.
think?
Yes,
So
sir.
nothing is bad over there. Look, but the only thing which is missing, the diagram. If the flow chart would have been there, I think she might have given you 2.5 at least for 3 marker question.
Okay, sir.
And if a teacher is giving 3 mark in your school, if teacher is giving 2.5 out of the three out of three, that's good, because
Yes,
sometimes
sir.
teacher do not give prefer to give like 100 out of 100.
Full marks, yes.
Yeah, but I have
I have
seen, like, yeah, I have seen the three girls in Delhi. Uh, I think last year or last to last year, they got 100 out of 100 in CBSE exam in biology.
oh,
And yeah, I was very happy. 100 out of 100 in biology, they got it, and I was happy, very happy. Not like this, because they got 100 out of 100. I proud on them, because it was not mathematics, physics, or chemistry where you could
yes,
get
sir.
100 out of 100. To get 100 out of 100 in biological sciences or life sciences or in humanities, that is really very Herculean task.
Yes, sir.
So you're in right way. The your handwriting is very good, and your content is like as per my.
So I should just work more on the diagram.
Yeah, the only thing that you need to represent it, and still
Okay, sir.
if you want the answer from my end, like if you want me to write
Yes.
down any answer for you, I can do that with a structure. I
Okay,
can do
sir.
that.
Okay, sir.
Yeah. So
Thank
someday
you.
I'll write answer with my own handwriting, and I'll upload over
Yes,
here.
sir.
Right. I'll give you people for
Okay,
all
sir.
of you. Right. And do that. Okay, Martin. Got it?
Yes, sir.
Yeah. You
Yes.
have to work hard. You have to work hard on answer writing, Martin. Okay. Every time, whenever I get the copy, first I check your copy.
Oh, okay.
Yeah. Next time I want a very good result from your end. Right. And what I will do, I will write a model answer with my own handwriting. I will scan it, and I'll upload you, right? And that's how I will tell you how to write a standard answer. Okay. So Shidika, did you got your query? Is it?
Yes, sir, I understood. I'll work on doing the diagram
Yeah,
more and separating my content.
exactly. And I'll within a day, I'll write an answer, standard answer, my own handwriting,
Yes,
and I'll
sir.
tell you how to write it down. Right.
Okay, sir.
Fine. So we were in plant physiology part, and we have to start from yes. Mhm. No, no, concentration. Where were we? we were, yes, we were here. We were transporting. Yes, yes, fine. So, Martin, did I dictated this one? Osmotic potential and all that last class?
Yes.
Okay,
Yeah.
fine, fine. So, Shidika, after the class, go through with it because this concept is a bit, uh, I won't say that this is difficult, but you need to understand it, right? A bit technical. Okay, so go through with the previous class
Yes, sir.
because this is important. And what will be the question for you, Martin and Shidika, both of you? What can be the question, or which question can be asked at your school? Either they will ask you osmosis for 2 mark, 2 mark or 3 mark. If they ask osmosis for 2 or 3 marks, you have to make the diagram. Okay. They can ask you one more thing. What is water potential? They can ask you this question as well, right? It will also be a 3 marker question, right? But the long question which they will ask you, they can ask you guttation for 2 marks, right? But what they can ask you as a long answer 5 marker question. The 5 marker question either will be transpiration pull theory or it will be mass flow theory, right? Mass flow theory. So that can be asked. I'll keep on telling you what can be the question for you. So what we did this one. Now let's start about the plasmolysis. What the plasmolysis is? Plasmolysis doesn't mean that a cell died, right? Plasmolysis is a process when cell gets shrink or uh, or it changes its structure, right? Its turgidity, it loses its turgidity, right? So this is a normal cell, right? In normal cell, what happened? I have already explained what is the hypertonic and hypotonic solution. Hypertonic solution means comparatively this is hypertonic solution means comparatively this is a concentrated solution. Hypotonic solution means comparatively this is a less concentrated solution. Now look, uh, when we keep a cell in a hypertonic solution means in concentrated solution, so what will happen? The water and minerals are stored in a cell inside where central vacuole, right? So we know that vacuole is the largest organelle of the cell, and when vacuole occupy lots of space at the center, the protoplasm and nucleus become peripheral, right? In any plant cell. Then what happened? So this is central vacuole, and this protoplasm, this green color is protoplasm. It is peripheral, and this is a nucleus, which is also a peripheral. And in the protoplasm, there are some small these organelle are present. Now, what will happen? Now this vacuole have pushed the protoplasm and nucleus aside. When we keep this cell in hypertonic solution, the water will start coming out from the central vacuole to outside, right? When water will come, this cell will shrink, become a bit flaccid, right? It will lose its turgidity. We will call it limiting plasmolysis. It's a very normal phenomenon. Why? Suppose that if you do not give water to a small plant for the next 2 days, you will feel that its leaves are dropping down, right? So that is a limited normal or limiting plasmolysis. Then the second stage, if further do not give you water, what will happen? Now, further, if you keep the cell in concentrated solution, what will happen? In next stage, incipient plasmolysis. What will happen in incipient plasmolysis? The protoplasm will start pulling itself from the corner of the cell, from the corner of cell, you can see, and it will start shrinking more. Vacuole will shrink more, and it will start pulling down. Means the contact between the protoplasm and the cell wall, right? Contact between the protoplasm and the cell wall gets separated in the corner. This is called incipient plasmolysis. Now further, if you do not give the water or do not pull out this cell from the hypertonic solution, what will happen? Except few places like this one, this one, except a few places, protoplasm will pull back itself and shrink and leave the cell wall, right? And between this space between the this protoplasm and cell wall, there will be hypertonic solution. Hypertonic pool solution will go inside the this space, right, and start filling over there. This is called evident plasmolysis. Finally, if you do not remove the cell from that hypertonic solution or do not keep it in the hypotonic solution, what will happen? This protoplasm will shrink and vacuole will become very small, and protoplasm will leave the cell wall and become this globular structure, right? And this hypertonic solution will get filled the space between the protoplasm and the cell wall. This is called final plasmolysis. So we can see that plasmolysis is of four stages: limited, incipient, evident, and final plasmolysis. During the limited plasmolysis, the cell becomes flaccid, right? Second stage is incipient, third evident. What is a deep plasmolysis? And even in this stage, if you keep the cell immediately after plasmolysis, if you final plasmolysis and keep the cell in hypotonic solution, cell will get its original stage, right? But for a limited time period, it will become alive, else it will die, right? So the process of keeping after the plasmolysis, when you keep the cell in a hypotonic solution, we call the process deplasmolysis. Means that is the opposite of plasmolysis. Is that clear?
Yes,
Did you got all
yes, sir.
stages?
Yes, sir.
Okay. So first, please put the heading plasmolysis. First, I will dictate, but after the class, you have to draw this diagram. And even if you want, you can draw right now, but I'll suggest you after the class, please draw the diagram, right? This diagram. So start writing now. Plasmolysis. Shrinkage of protoplast of a cell. Shrinkage of protoplast of a cell from its cell wall under the influence of hypertonic solution, under the influence of hypertonic solution is called plasmolysis. Full stop. Hypertonic solution causes exosmosis. Do you know exosmosis and endosmosis? I told
No,
in last
sir.
class. Okay,
I
I have told
forgot.
in last class. Let me tell you what is exosmosis and endosmosis. Look, when in osmosis, where is it? Yes. In osmosis, last class, I told to Martin, that osmosis is of two types. Endosmosis, when water come inside the system. For example, you have a resin, put that resin. So resin is shrink kind of structure. When you put this resin in a glass water overnight, at morning you will see it have swell and it look like a grape, right? So the water goes inside the system. That is called endosmosis. What is exosmosis? Take a grape and put this grape in concentrated salt water. After some time, you'll see that the water is coming out of the grape. When it came out of the grape and grape have shrink. This is called exosmosis. Got it?
Yes, sir.
Okay. Then that is exosmosis. Fine.
Yes, sir.
What you written? Shrinkage of protoplast cell under the influence of hypertonic solution is called plasmolysis. Okay. Hypertonic solution causes exosmosis or withdrawal of water from cytoplasm and then the central vacuole of cell. Then the central vacuole of cell. Full stop. The size of cytoplasm as well as central vacuole as well as central vacuole become reduced. Full stop. The pressure on the wall is subsequently reduced and elastic wall contract, causing a reduction in size and causing reduction is in size. Full stop. The first stage of this first stage of plasmolysis is called limiting plasmolysis. Full stop. The cell is called flaccid. The cell is called flaccid. Next paragraph. The extra hypertonic external solution continue to withdraw the water from central vacuole. Full stop. Initially, the protoplast withdraw itself from corners. This stage is called incipient plasmolysis. Full stop. The hypertonic solution now enters the cell in between the protoplast and cell wall. Full stop. Next paragraph. Due to continued exosmosis, protoplast shrink further and withdraws from the cell wall except one or few places. Full stop. It is known as Evident plasmolysis. Evident plasmolysis. Next heading is deplasmolysis. Deplasmolysis. The swelling up of a plasmolysed protoplast under deplasmolysis. Swelling up of a plasmolysed protoplast under the influence of hypotonic solution. Under the influence of hypotonic solution or water is called deplasmolysis. Full stop. It is due to endosmosis. It is due to endosmosis. Full stop. Deplasmolysis is possible. Deplasmolysis is possible only immediately after plasmolysis. Only immediately after plasmolysis. Is that okay, everything? Shritikant, Martin, both of you?
Yes, sir.
Okay. At least you have to draw these three diagrams: limiting, incipient, and evident after the class. Okay. Whenever you get a question from the plasmolysis, you must have to draw this diagram, right? Else they will cut down your marks. Fine.
Yes.
Okay. Now come to the long distance transfer. Martin, did you got everything in plasmolysis?
Yeah, yes.
Okay. Okay. Fine. Give me just 1 second. So then a long distance transport, right? So what is the long distance transport? Mhm. What is long distance transport? Can you tell me? Yes. Any idea what is the long distance transport?
Nothing.
No. Martin, what's about
I
you?
don't know, sir. No, I don't.
You don't know. Okay. So let me tell you what is the long distance transport. Long distance transport means when you are transporting something for longer distance. So short distance transport, when I started teaching you, when I was teaching you, Martin, diffusion, then I said diffusion is possible in one cell, two cell, or in surrounding cells. That is the only thing where the diffusion is possible, right? But diffusion cannot go for the 10 centimeter or not for the 1 meter. But the plant can be of 4 meter, 5 meter, 10 meter, right? The length of a sequoia plant can be up to 200 meter. So 120, sorry, 120 meter. Yes, 120 meter or 1111 meter. Eucalyptus plant can be of 111 meter. So how can a water can go in that much height from root to the top? That is a long distance transport. So this is possible only with the help of long distance transport. So when water is transported in a plant, it may be 10 feet, 20 feet, 15 feet, 50 feet, or 60 feet plant in general. So, but you know that in 50 feet or 60 feet, even in 100 feet, so to take the water at the top of your building, you require a water motor and you require lots of pressure. Then only the water can reach up to the top of your building. That's how you fill your tank, right? But the plant do not have that kind of the motors. And how does the plant transport water? Look, so the transportation of water through a in a plant is possible only with the help of long distance transport. So long distance transport, epidermis. First, let me tell you. So it starts from the root to the top. That is called long distance. In the same way, the second thing, what is transported in for the long distance, that is food. How? Root are doing their job. They are providing water and mineral to the plant, but in return, they are not getting anything, right? But they require something which they cannot make. What they cannot make, uh, that is a food. So how does they get the food? Actually, the leaves then do their job. In return, they give something because they are getting the water, right? Root is giving water to the leaves. In return, leaf give back food to the root. How the food is transported from the top of the plant to the bottom of root? That is with the help of phloem, and it also travel from the top to the bottom. That is also long distance transport. So in long distance transport, we study two things: transport of the water and transport of the food, right? For transport of the water, try to understand what exactly I want to say you. Look, for the transport of the water, there is a theory that has been given. It is there in your book, right, in long distance transport. So transport of the water is uh, explained by a theory that is called transpiration pull theory. This theory, transpiration pull theory, which we will study, this theory is explaining how does water is transported from the root to the top of the plant. Now, the second thing which I said to you, long distance transport, that is transport of the food. How does the transport of the food is done? Transport of the food is done with the help of a theory that is called mass flow theory, right? Source and sink theory, this mass flow theory, right? So this is mass flow theory, source or sink theory. This explains how does the food is transported. So these two theories are important for you as a 5 marker or long answer. Got it? Got it or
Yes,
not?
sir.
So look, long distance transport, in long distance transport, uh, anatomy we have already understood. Look, let me tell you, this is the structure of the root, right? So in root structure, do you remember this structure? The outer layer is called epidermis, root hair or epidermal hair, remember or not?
Yes, sir.
After epidermis there is a cortex, Martin, remember?
Yes.
The innermost layer of the cortex is endodermis, and then after there is a vascular system, xylem or phloem. Yes. So water and mineral, they are absorbed by the root hair. From root hair they reaches to the cortex. From cortex they reaches to the endodermis. From endodermis they reaches to the xylem or phloem. Okay, so that's how it travels. Now come to this diagram. This diagram is already there in your NCRT, and uh, you can get this diagram from there as well. And this is important. There are two paths of the flow of, try to understand, there are two paths of the for the water to travel inside the plant cell, right? Symplast and apoplastic. Look, water, so this outer layer is epidermis, then this is cortex, then there is endodermis, and then there is a pericycle. In endodermis, there is a thickening of some material, and we call that Casparian strip. Remember Casparian strip?
Yes, sir. Yes, sir.
Casparian strip do not allow the passage of water. In between, there are some cells called passage cell. These passage cell allow the entry of the water, right? Look, when water enter to endodermis, then epidermis, water enter into the protoplasm of cell wall. From protoplasm, one cell between the two cells, there are some pores or correction. They are called plasmodesmata. From epidermis, it enters into the uh, protoplasm of cortex. From cortex, it enter into endodermis. From endodermis, then it enter into pericycle, and then into xylem or phloem. Right? That's how the water travel from one point to another point. Right? Then what happened? So this is here. What is going on? Look, try to understand here. This is the external part, that is a cell wall, right? Then there is a plasma membrane, and then there is a protoplasm, and inside there are vacuoles. So what happened? First enter water, enter into protoplasm, then into vacuole, from vacuole then into protoplasm, and then into plasmodesmata to next vacuole. That's how the water travel in the symplastic pathway. Now, what's about the apoplastic pathway? In apoplastic pathway, there is a gap between the plasma membrane or a protoplasm and the cell wall. This gap is called apoplastic pathway. When water travel in the gap between the protoplasm, it do not enter into vacuole or it do not enter into protoplasm. When water do not enter into protoplasm, this is called apoplastic pathway, right? So here there is a gap. These dark pink structures are apoplastic pathway. So in epidermis, say water enter in symplastic as well as apoplastic pathway. Then come to the cortex. Even in cortex cell, water travel through the apoplastic and symplastic both pathway. But you know the Casparian strip do not allow the water to flow through the apoplastic pathway. Now in Casparian strip, only symplastic pathway is possible. So when water reaches to Casparian strip, it follow the symplastic pathway. After that, it enter into pericycle, symplastic pathway, then reaches into xylem, right? So these are the two pathway, apoplastic and symplastic. You will see that here and separate diagram, I have shown both of the thing with the help of a separate diagram. When symplastic pathway, you can see the water is passing through the space between the protoplasm and cell wall. In symplastic pathway, the water is entering through the protoplasm. So these are two kind of the pathways, symplastic and apoplastic pathway. Tell me guys, is it clear or not?
Yes, sir, it's clear.
Clear? Okay. Take this screenshot, make the diagram. Diagram is mandatory. Look here. This blue color is symplastic pathway, red color is apoplastic pathway. Root here, then into cortex, right? In cortex, this is symplastic pathway. Look, when it came into here in this endodermis, right? Then it followed the symplastic pathway. Even for the this one pink color, apoplastic pathway, apoplastic pathway, apoplastic. When it came, it reached to the endodermis, it had to follow symplastic pathway, right? So in endodermis, apoplastic pathway not possible. In rest of the cell, apoplastic pathway is possible, right? Done. So
Yes,
put the
sir.
heading. Put the heading. But main heading is long distance transport, right? And start writing. Sorry. There are two pathway of water passes. There are two pathway of water passes from root hair to xylem. From root hair to xylem. Inside the root, apoplast and symplast. Apoplast and symplast. Full stop. First subheading, apoplast pathway. Small heading, subheading, apoplast pathway. Here, water passes from root here Water passes from root hair to xylem through the walls of intervening cells. Without crossing any membrane or cytoplasm. Full stop. The pathway provide the least resistance to movement of water. Pathway provide least resistance to movement of water. Full stop. Next point. However, it is interrupted by presence of Casparian strip. However, it is interrupted by impermeable Casparian strip. Impermeable Casparian strip. Full stop. Next, subheading symplastic pathway. Next subheading is symplastic pathway. In symplastic pathway, water passes from cell to cell. Water passes from cell to cell through their protoplasm. Full stop. It does not enter the cell vacuole. Full stop. The cytoplasm of adjacent cell are connected. The cytoplasm of adjacent cell are connected. The cytoplasm of adjacent cell are connected through bridges called plasmodesmata. Full stop. For entering into symplast, water has to pass through plasmodesmata. And cell membrane. Full stop. One last point. Symplastic pathway is added by cytoplasmic streaming. Do you know what is cytoplasmic streaming? Do you know? Edukiran Martin, do you know what is cytoplasmic streaming? Tell me, Martin.
Yes, sir. Yes.
What is cytoplasmic streaming? Look, cytoplasm keep on rotating or revolving, right? Keep on rotating like this way, right? This is called cytoplasmic stream, right?
They keep on rotating.
Yeah, so keep on rotating and moving like this way, right? This is called cytoplasmic stream. It add on. in the protocol. It add on in the movement,
Okay.
right? In the last, you can write down one more thing. Both pathway are involved. Both pathway are involved in movement across the route. Next point. In the same paragraph, mineral nutrient also have mineral nutrient also have the same pathway as that of water. Mineral nutrient also have the same pathway as that of water. Full stop. However, their absorption and passes, however, their absorption and passes into symplast mostly occur through into symplast mostly occur through in the symplast mostly occur through active absorption occur through active absorption, right? Inside xylem, the movement is purely inside xylem, the movement is purely due to pressure gradient. Do you know what is the pressure gradient? Martin and Shidika, what is pressure gradient? From high osmotic pressure to low osmotic pressure. That's it. From high osmotic pressure to low osmotic pressure. Did you got this point or not? Martin and Shidika, tell me, please.
Yes, sir.
Yes, sir.
Okay, great. So this diagram you have to make, right? Fine. Then there is a one more thing that is called mycorrhiza. What is mycorrhiza? Any idea? Mycorrhiza, I have told you already. Now, mycorrhiza, did I told you or not? Tell both of you. Did I told you mycorrhiza or not? You will tell. Come on, Martin, Ritika,
Coriza. No, no, I don't think so.
you don't think so? Okay, fine. Ritika, what's about you?
Um,
Did we discuss
I,
about or not? No?
I don't think so, sir. Although I
Okay,
did hear this word in class, I
fine,
can't
fine.
recall it.
But we did the plant kingdom or not? We did it.
Yes,
Plant fungi, yes,
yes, sir.
and we did with same PPT. Let me check. Did we complete it? Mycorrhiza, we did it or graphite addition of generation? Okay, let's plant. This fungi. Fungi we did, yes. Where is it? Should be here. Okay. Let's put that well, classification of, or I think classification for, yes. There is the mycorrhiza we did not discuss. Okay. Lichen, lichen is a symbiotic relationship, maybe. Maybe I don't know where. I think mycorrhiza we have discussed already. Like in lichen, do you remember lichen? Hello, Siddhika and Martin. Remember lichen?
Yes. From the other animals, like in
Yeah. Yes, Martin. What are you saying?
another.
Look, lichen, we call them dual organism, right? There is a algal partner, there is a fungal partner, right? Algal partner we call phycobiont and fungal partner we call mycobiont, right? Uh, algae is green, it do photosynthesis, provide food, and in return, fungi provide what? Water and mineral. And this symbiotic relationship is called what? Lichen, right? In the same way, what happened with the plant, right? Mycorrhiza is the same kind of the thing. So what is the mycorrhiza? It is a mycorrhiza is also a symbiotic relationship, right? This is a symbiotic relationship between a plant and between a plant and fungi. Here there is a symbiotic relationship. Obviously, it's a plant and fungi. The same thing happened, but there the symbiotic relationship is with the higher plant, right? So look, what happened in mycorrhiza? Let me make you diagram. This is basically a pinus or cycas plant, means gymnosperm. So pinus and cycas plant, they have the root, but their root are not capable enough to take water from external environment. They are not that much efficient like angiosperm. So what happened around? So this is the root of pinus. You have to make the diagram as well. Root of gymnosperm like this. Gymno. So this is root of gymnosperm. Around the root of gymnosperm, they make a network of fungi. Right? So fungi make a network. This is a fungal, mental, or fungi network. Right? It make a network, and this is root of gymnosperm. What happened? Fungi provide gymnosperm two things. What? Efficiency to absorb water plus mineral. Right? What happened? What it give? In return, a root give to fungi what? Food. Right? So this is a symbiotic relationship between symbiotic relationship symbiotic relationship between whom? One, higher plant, higher plant and fungi. So between higher plant and fungi, there is a symbiotic relationship. This is called mycorrhiza. Clear? This is important. This is very important question. Mycorrhiza. Not important, very important. Make the diagram quickly. Come on.
Done, sir.
Done? Very good. So start writing now. In mycorrhiza, a large number of fungal hypha. In mycorrhiza, large number of fungal hypha are associated with young root. Full stop. The fungal hypha are extended to sufficient distance, extended to sufficient distance, sufficient distance into the soil. Full stop. They have large surface area. They have large subsurface area. Full stop. Hypha, hypha are specialized. Hypha are specialized to absorb water and mineral. Hypha are specialized to absorb water and mineral. Full stop. Uh, in return, root provide fungus. In return, root provide fungus, sugar and nitrogen containing compound. Full stop. Mycorrhizal association between fungi and root is often obligate. Full stop. Pinus, cycas, and orchid root. Pinus, cycas, and orchid root are the best example of mycorrhiza. Clear?
Yes, sir.
Okay. The next concept is guttation. No. In African continent, there are a tribe known Masai. Have you listened about this tribe, Masai? They're good hunters,
Okay.
Masai people. So Masai people, they are a good hunter, and they have been described in history in widely. And it's a very prominent tribe, Masai. They are very brave kind of people, and they are still nomad. So what they do? So they live around the Sahara Desert, in that area, means northern part of Africa. So in those desertic area, the water level is very down and water is very scarce. So how does they collect the water? They have a very at the surrounding where they live, they have some plants, and the leaf of the plants are very wide, right? They have wide leaf plants. So these wide leaf plant, at the tip of these wide leaf plant, there are some pores. These pores are known as the hida throat, right? And at the night time, when night become, in desert, days are very hotter and nights are very cooler, right? So in the daytime, transpiration is start very speed. Transpiration goes on very speedily, and in the night time, transpiration rate goes down. So what happened? Those plant have the pores at their tip. These pores are called hydathoid. Through them, the water comes down in the droplet, right? What these Maasai tribe do, they collect the ostrich egg shell. It's very hard, work like bowl. So they make the bowl of ostrich egg shell, and they keep these bowl overnight. And at the morning time, these bowl get filled, and they collect the water from this bowl, and then they collect this water and they use the water for the whole day for their requirement, for drinking purpose, right? That's how they collect the water. And at the daytime transpiration is very high and the nighttime transpiration do not take place, right? So the root pressure remains maintained. So what happened in the daytime when the transpiration rate is very high, so days are very hot and then the root pressure is very high, right? In the night time what happened, the transpiration rate become almost zero, but root pressure remains high. Due to this high root pressure, the water come out from the here because it cannot get transpirated, right? And these pores are called hydrothoid. There is a diagram, and this process of water coming out, water in the form of droplet, is called guttation, right? For guttation takes place through the hydrothoids. This is the anatomical diagram of hydrothoid. There are some loose cells. These cells are called epitheme. Epitheme is, what is epitheme? Epitheme is nothing but this is the loose parenchyma cell, right? And then you will see there is a guard shell like stomata, and these are trichate of xylem. So water droplets come out from these pores. Did you got this concept, whole concept?
Yes, yes.
Make the diagram, because diagram is not there in your book. Make the diagram and make the diagram of hydrothoid as well. Not that much complex. Make a rough diagram like, I mean, a easy diagram. Make, do not copy it as it is.
That's it.
Done. Okay. Now note down what is the guttation. What guttation is. Yes, please note down. The loss or excretion of water, the loss or excretion of water in the form of loss or excretion of water in the form of liquid droplet, liquid droplet from the leaves or other part of or other part of an in or uninjured an uninjured or intact plant is called guttation. Full stop. All plant do not show guttation. Example,
All plant do not show.
guttation, right? Example, for example, you can note down. Example, tomato, tomato soup, tomato, right? Cucurbits, this pumpkin, cucurbita, right? Pepo, oats. There is a plant which is very popular, garden nasturtium. So you can note down. Don't forget this one, garden nasturtium. Garden nasturtium, right? Tomato, cucurbit, oats, and garden nasturtium. They are the example of guttation. Next point. The gutated liquid is never pure water. We do not drink pure water, we require the mineral water. The gutated liquid is never pure water. It contain both organic and inorganic salt. It contain both organic and inorganic salts and compound. Next point. Guttation usually occur during the period of guttation usually occur during the period of active growth when conditions favor more water absorption and less transpiration. Full stop. Such conditions are found in humid tropical regions. Next point, next paragraph. Guttation takes place through a special structure, either thought. Guttation takes place through a special structure, either thoughts. They are usually found in the margin of tips of the leaf. That's it. So this was all about the guttation. And then transpiration pull theory, no, I will not discuss today. Look, what is a transpiration? I think you must have fair idea. What is the transpiration? What is transpiration? Tell me quickly. Martin and Chilika, both of you.
Is a
What is transpiration?
loss of water in the form of vapor.
Yeah, exactly. Loss of water from the plant is called transpiration, right? And what is the type of transpiration? Here we can discuss. The first transpiration is stomatal transpiration. We know that stomata are small pores which are found more on the lower surface of leaf, right? So the evaporation or losing of water uh, from stomata is 50 to 97%. So 50 to, suppose that there is a plant which every day loses 10 liter water. So out of that 10 liter, 50 to 97% will transpire through stomata, right? Then in the stem and rest of the bark part, outer skin part, right, means epidermal part. So there is an epidermal part which is covered with the cuticle, right, a waxy layer. So 3 to 10% that evaporate from here. This is called cuticular transpiration, right? Because other than stomata, the skin is covered with the cuticle. Leaf skin is covered with cuticle. Cuticle is waxy substance. Still, it is not completely impermeable for water. 3 to 10% water loses through the cuticle, right? So we call it cuticular transpiration. Then in woody plant, in the stem of the plant, there are some small pore which remain open permanent, right? They're very small pore which are found in the stem, but they remains open for the throughout the lifetime, right? So these small pores are called lenticels. So that we call lenticular or lenticular, right, transpiration. Lenticular transpiration is 0.1% of all transpiration. Then the fourth one is bark transpiration. From the bark, some amount of the water also transpires through the bark. This is called bark transpiration, right? And this is also goes on day and night. And this is basically in the corky covering of a stem. This bark is corky covering of a stem, and through the bark transpiration takes place. This is also called transpiration, right? So there are basically four kind of transpiration: stomatal transpiration, lenticular transpiration, uh, lenticular transpiration, and bark transpiration. Clear? So you can note down the percentage with the type of transpiration, and in next class you have to put the heading. We have to start from the uh, transpiration pull theory. I will start this theory. Transpiration pull theory. This theory I will start. Right. So note down for this stomatal, cuticular, and lenticular transpiration. We'll keep today till this point.
Done, sir.
Done. Okay. Done, Martin.
Yes, sir.
Okay. So see you next class day after tomorrow, and we'll study from the transpiration pull theory. Right. Bye bye. Take care. Good night.